Good day, everyone. I am Jaira Cedar Pisa, and today, together with my group mates, we will be presenting an important topic under VetMed 529A, Aquaculture and Aquatic Management. This is the feeds and feeding management. Proper nutrition and feeding strategies play a vital role in the growth, health, and productivity of aquatic species, making this topic essential for efficient aquaculture practices. Before we dive into the specifics, let's take a moment to understand why feeds and feeding management are crucial in aquaculture. Just like any other animals, aquatic species require a balanced diet to thrive. The efficiency of feeding not only impacts growth, survival rates, but also affects water quality and production costs. With that in mind, let's go over the objectives of this discussion. By the end of this discussion, you should be able to identify and understand the importance of different nutrients required by aquatic species, formulate feeds using locally available indigenous feedstuffs, recognize various feeding strategies and apply proper feeding management techniques, and evaluate fish growth using different methods of analyzing growth parameters. Now that we have established our objectives, let's begin by exploring the fundamental nutrients required by aquatic species. Fish require proper nutrition for growth and health. Their food is classified into two types, the natural foods and the artificial foods. These are natural present in water, such as plankton, algae, and detritus, is the natural food, and the artificial foods are provided to reared aquatic animals to supplement or replace natural food. Kinds of artificial feed. Single ingredient feed. These are from the plant origin such as soybean meal, rice bran, and corn, and the animal origin such as fish meal, shrimp meal, and blood meal. Meanwhile, the compound feed is a mixture of two or more ingredients for balanced nutrition. Artificial diets are classified based on nutrient composition and physical form. Classification of artificial diet or feed. First is the purified diet. It contains pure nutrients such as casein, which is 99.99% protein. Then the practical diet, which contains varied nutrients from a single ingredient, such as fish meal, or a compound feed, such as commercial pellet feed. Next is the physical form of feed or diet. First, the pellet, which is tubular-shaped feed. Crumble, which is broken pellets with irregular shapes. Powder or mash form, finely ground feed. Flakes or thin flat feed pieces. The granulized, which is small uniform granules. Ball form, which is rounded feed pieces. And the cube form, which is compact cube-shaped feed. Pelleted feeds are classified based on their buoyancy and feeding behavior of fish. There are two kinds of pellets, the sinking pellets and the floating pellets. Sinking pellets are ideal for bottom feeders and can also be consumed by column and surface feeders in low water levels. Meanwhile, the floating pellets are best for surface and column feeders, allowing easy monitoring of feed intake. Next is the physical condition of feed or diet. The dry feed. It contains less than or equal to 10% moisture content. The moist to semi-dry feed, which has 30 to 40% moisture content, and the wet feed contains 46 to 70% moisture, such as the trash fish or the fresh mussel meat. Feeds are classified based on the growth stage of aquatic animals with varying protein levels. The type of feeds are the following. Pre-starter feed, the highest protein content formulated for early growth stages. Second is the starter feed, which supports initial growth and development. Next is the grower feed, designed for the rapid growth phase, and the finisher feed, which has the lowest protein content used before harvest. The following are the brand names of feeds: the BMEG, the Vita-Rich, Tate, and AQUA, and others available in the market. Apart from choosing the right type of feed, proper feed formulation plays a crucial role in ensuring optimal nutrition for aquatic species. The feed formulation. Feed formulation is the process of mixing various feed ingredients, vitamin and mineral supplements, and additives in the right proportions to create a diet with the required nutrient and energy. The major factors governing the choice of feed ingredients. First is the suitability for the species. Ingredients must provide essential nutrients, be highly digestible, and have good palatability. They should also be free from adulteration and toxins. Next is the quantity and regularity of supply. Choose ingredients that are consistently available in sufficient quantities. Last is the cost. Select cost-effective feedstuffs based on the price per unit weight of protein. Example, assorted fish meal, which has 55% protein at 22 pesos per kilogram. Next is the anchovy meal, which is 70% protein at 27 per kilogram. Which one is more economical? Let's compare two feed ingredients, the assorted fish meal and the anchovy meal. For the assorted fish meal, we divide the cost per kilogram of the ingredient by its protein percentage. So 22 pesos divided by 0.55 equals 40 pesos per kilogram of protein. Meanwhile, for the anchovy meal, applying the same formula, 27 pesos divided by 0.70% equals 38.55 pesos per kilogram of protein. Although anchovy meal costs more per kilogram as an ingredient, it provides protein at a lower cost per kilogram, making it the more economical choice. Balancing a feed ration ensures that aquatic animals receive the necessary nutrients in the correct proportions. There are three main methods used. These are the following: the Pearson square technique, the multiple or trial and error method, and the algebraic method. So the Pearson square technique is a method that calculates the exact quantities of ingredients needed to achieve a specific protein level in feed formulation. For example, for diet with two ingredients, formulate a diet to contain 25% protein using fish meal with analyzed crude protein content of 53.4% and a fine rice bran with analyzed crude protein of 12.9%. To ensure proper nutrition for aquatic animals, we need to balance the feed ration correctly. One way to do this is by using the Pearson square, which helps us calculate the exact amount of each ingredient needed to achieve the desired protein level. Let's say we want to make a diet containing 25% protein using two ingredients. So we place the target protein level, 25%, in the center of the square, with the protein content of each ingredient on the sides. So we have the fish meal, which is 53.4% crude protein, and the fine rice bran, 12.9% crude protein. Then the target protein level is 25%. So step 2, subtract to get the differences. We need to subtract the target protein level from the protein content of each ingredient to determine their respective contributions. For the fish meal contribution, we have 53.4% minus 25% equals 28.4%. For the fish rice bran contribution, 25% minus 12.9%, which yields to 12.1%. And the step 3, find the percentage of each ingredient. We need to calculate the percentage of each ingredient in the final mix. 12.1% over 40.5% times 100 equals 30% of the fish meal, and 28.4 over 40.5 times 100 equals 70% fine rice bran. To achieve 25% protein diet, we mix 30% of the fish meal and 70% fine rice bran. Although fish meal has a high protein content, we only need a small amount of it, while larger portion of the fine rice bran helps balance the mixture and provides energy. Now, let's look at another example using three or more ingredients where specific percentage, shares, or parts are designated in the formulation. For example 2, formulating a 25% protein diet with multiple ingredients, we will use different protein sources and calculate their contribution to the total protein content. Given ingredients, in Group 1, the protein sources, which are the fish meal, which is 59.8% crude protein, the soybean meal, which is 47.5% crude protein. Step 1, multiply each ingredient parts by its protein content. So fish meal is 1.5 parts times 59.8% crude protein yields to 89.70%. The soybean meal, which is 2.5 parts times 47.5% crude protein, equals 118.75% protein contribution. So the total parts of this is 4 and a total of crude protein of 208%, 208.45%. Next step, adjust to target protein level. The total protein contribution from both ingredients is 208.45%. From here, we will adjust by adding energy sources or other feed components to balance the diet to the required 25% protein level. So, by using multiple ingredients, we ensure a well-balanced diet with the right protein levels for optimal fish growth. Now let's complete our formulation by incorporating energy sources and calculating the final balanced protein. So first step, calculate the weighted average for protein sources. From our previous calculation, the total crude protein contribution from protein sources, which is the fish meal and the soybean meal, is 208.45%. To find the weighted average, is 208.45% divided by 4, which yields to 52.11% of protein. Then step 2, calculate the weighted average for energy sources. Now let's compute the contribution from energy sources, which is the rice bran and the wheat flour. So rice bran is equal to 3 parts and 9.8% crude protein. So 3.9.8 equals 29.40% protein contribution. For the wheat flour, 1 part times 14.2% crude protein yields to 14.20% protein contribution. So the total parts is 4% and the crude protein is 43.60%. So 43.60% divided by 4 equals 10.90% of protein. So let's proceed to the final feed formulation using the percent square. We balance the feed using the percent square technique to achieve our target protein level of 25%. So for the protein source, weighted average is 52.11%, and the difference is from the target protein minus 25 equals 14.10. So the 14.10 divided by 41.21 times 100 equals 34.22. Then in the energy source, 10.90%. 25 minus 10.90% equals 27.11%. Now we will be dividing that to 41.21% times 100 equals 65.78%. Now that we have a total of 100% in the percentage and the protein is 41.21%. So the 34.22% protein sources, which are the fish meal and the soybean meal, and the 65.78% energy sources are the rice bran and the wheat flour. So to achieve a balanced diet with 25% protein, we mix 34.22 protein sources and 65.78 energy sources. Another method for balancing feed rations is the multiple or trial and error method. This approach involves summing up the protein contributions of different ingredients and adjusting their inclusion rates until the desired protein level is achieved. For example, formulating a 25% protein diet. The trial one, or the initial attempt, in our first trial, we include fish meal, soybean meal, and rice bran with the following calculations. So the inclusion in rate in the fish meal is 20 grams and the crude protein is 53.4%. So the protein contribution is 20 times 53.4 over 100 equals 10.68% protein contribution. And for the soybean meal, 20 grams and a crude protein of 45.9, 20 times 45.9 over 100 equals 9.18%. And the rice bran, which includes 60 grams and 12.9% crude protein. 60 times 12.9 over 100 equals 7.74. With a total of 100 grams, we have a 27.60% crude protein, which is above our target. So, adjusting the inclusion rate since the total protein content is too high, 27.60% instead of 25%, we modify the proportion of the fish meal and the rice bran to bring it closer to 25%. So, the fish meal from 20 grams, we will be making it into 40 grams. It has a crude protein of 53.4%, so 53.4% over 100 times 14 grams equals 7.476%. And then the soybean meal, which will be remaining as 20 grams and a crude protein of 45.9%, 20 times 45.9 over 100 equals 99.180%. And the rice bran from 60 grams, we will be making it to 66 grams. So with a crude protein of 12.9, 66 times 12.9 over 100 equals 8.514%. So with a total of 25.17% protein contribution, we achieved the target 25%. Through trial and error, we successfully adjusted the ingredient proportion to achieve the desired 25% protein level. So this method is practical and flexible, especially when working with multiple ingredients, but it requires repeated adjustments to meet the exact protein requirement. Now let's move on to the evaluation of feeds, which is crucial in ensuring quality and efficiency in aquaculture nutrition. There are four major ways to evaluate feedstuffs and feed formulation: the physical evaluation, biological evaluation, the chemical evaluation, and the microbiological evaluation. The physical evaluation. The physical characteristics of feed can be assessed using two methods, the macroscopic observation and the microscopic examination. The macroscopic or gross observation. This method relies on our senses to assess feed quality. The sense of smell, which detects rancidity or foul odor. Sense of taste identifies off flavors in feed. Sense of sight, helps spot mold, insects, contaminants, and assess compactness, and sense of touch determines texture, hardness, or coarseness of the feed. Second, the microscopic examination. For a more detailed evaluation, microscopic analysis is used to check for starch content, bacterial or mold contamination, and moisture levels. Physical evaluation is the first step in assessing feed quality, ensuring that it meets the standard for safe and effective feeding.
The second evaluation of feed is the biological evaluation. So, biological evaluation is a way of evaluating the effect of the food or the feed on the animal itself. So, we have two parameters used to evaluate the effect of feed on the animal. First, we have the direct parameters, and these are growth, survival, tissue, and skeletal changes. For the indirect parameters, we have feed conversion ratio or feed efficiency. This is for food consumption. Protein efficiency ration for protein consumption, and we have the productive protein value or apparent net protein utilization. This is for protein utilization. So these are the measures used in evaluating the feed or the food under biological method. So for the growth, we can use any of the three methods that we can find in the reference. To find the daily weight gain, the formula is mean final weight minus mean initial weight divided by the culture period, that is in days. For the next method used in evaluating the food or feed, we have for food consumption. We can also use any of the formulas used in the reference, and one of them is the food conversion ratio, which is defined as the weight of feed ingested in gram per gram gain in weight of fish. So the formula for that is food ingested in gram or food eaten divided by gain in fish weight or final fish weight minus initial fish weight. For the protein consumption, we can use the protein efficiency ratio, which is defined as the gain in weight of fish in gram per gram protein consumed. So this is the formula: gain in fish weight in grams divided by protein intake. For the protein utilization, we can use the apparent net utilization or ANPU, which is defined as the percentage of ingested protein which is deposited as tissue protein. So the formula for this is PE minus PS divided by PI times 100. To get the PI, we have the total feed given multiplied by the percent crude protein of feed. PE is the weight of fish body protein at the end of the feeding, PS is the body protein weight before the start of feeding, and PI is the weight of protein consumed over the feeding period. For the next evaluation of feed, we have the chemical evaluation. This is a method of evaluating feed which determines the amount of the 6 feed fractions or components, such as moisture, crude protein, crude lipid or fat, nitrogen-free extract, crude fiber, and ash or the mineral matter contents, through a system known as the proximate analysis. This method also determines the other nutrients that the feedstuff or diet itself contain. The following are the methods of determining the 6 feed fractions or components. For the moisture content, it is obtained by oven drying the feedstuff or diet at 105 degrees Celsius until constant weight is obtained. The formula for this is loss of weight during drying divided by weight of sample before drying times 100, or for the percent of dry matter, we have weight of sample after drying divided by weight of sample before drying times 100, and then percent of moisture content is equal to 100 minus dry matter. For the next method, we have the crude protein content. This is determined either by macro-Cheldahl or Cheldahl method, which estimates the total nitrogen content, which is in turn used to determine the quantity of protein by multiplying the natural content obtained with 6.5. Another, we have the chemical score, which is a method used to determine quality of protein. This is done by comparing the essential amino acids in fish or feed protein with that of the whole egg protein. Next, we have the essential amino acid index. It is also for determining the quality of protein. If the EAA, or the essential amino acid required by fish, is not known, then compare the EAA of the diet to the species mussel to be fed. For the next method for this chemical evaluation, we have for the crude lipid content. This is determined either by the Folk method. Lipid is extracted by blending procedure using 2:1 ratio of chloroform and methanol respectively. Bly and dryer for the lipid extraction using first blending, that is 1:2:0.8 chloroform, methanol, and water. The second blending for the bly and dryer is 2:2:0.8 chloroform, methanol, and water, and the third or the last blending is 2:2:1.8 chloroform, methanol, and water. Next is the ether extract. It is a traditional method of extracting lipid with ethyl ether in Soxhlet apparatus. The next method for the chemical evaluation is for the crude fiber content. This is determined by initially washing the organic residue that remains after boiling the sample material with sulfuric acid solution, sodium hydroxide solution, and ethanol in this sequence. Finally, by drying the washed defatted sample in an oven at 105 degrees Celsius until constant weight. For the next method of the chemical evaluation for the nitrogen-free extract, it is determined by the formula 100 minus the sum of moisture content plus crude protein plus crude fat plus crude fiber plus ash. Now for the last method of the chemical evaluation for the ash, it is analyzed by incinerating the fiber content in furnace at 550 degrees Celsius to 600 degrees Celsius until carbonaceous matter is oxidized. Now for the last method of the evaluation of the feed, we have the gross energy. Gross energy of a feed or feedstuff is determined with a bomb calorimeter. It is the total heat or combustion of a feed being analyzed. It is usually expressed as kilocalories per kilogram of feed. We have here a table of the factors used for calculating the digestible energy and gross energy of feed or feedstuffs using the following calorific values. In calculating the digestible energy value of a feed or feedstuff, the only components considered are protein, lipid, and nitrogen-free extract. But before DE, or the digestible energy, is calculated, such components must first be converted to a dry matter or moisture-free basis. Now for the next part of my report, we have the feeding strategies. So we have 4 feeding strategies, and these are: first, feeding with naturally grown lab plankton or lumot without any fertilization. The next, we have the same feeding strategy as the first, but with the use of organic and inorganic fertilization. Next, we have the feeding with naturally grown food plus supplemental feeds. Lastly, we have complete feeding and ad libitum feeding. So lab is a kind of microbenthos composed of blue-green algae, diatoms, and other microscopic plants and animals. Lumot is composed mostly of filamentous algae such as Chaetomorpha, which are also growing in milkfish ponds. We have here an example of an organic fertilizer. This fertilizer is used for the growth of plankton. Supplemental feeding, by definition, is the feeding with supplemental feed to meet the nutrient requirement of fish for maintenance and growth when natural food is inadequate. Usually, supplemental feeding is given 1 month after milkfish fingerlings are stocked, when their biomass is about 300 to 400 kg per hectare at the rate of 2 to 4% of fish body weight when natural food is low, and at 1.5 to 3% when these are abundant. Supplemental feeds could be in the form of formulated pellet feed, that is commercial or homemade, and single ingredient feed like the rice bran, breadcrumbs, or any other natural or artificial foods. For the feeding frequency, It is 3 times a day when natural food is scarce and feed is given at 9 to 10 a.m., 12 to 1 p.m., and 4 to 5 p.m. The total feed for the day will be divided into three, of which 25%, 40%, and 35% of such daily feed will be given to each feeding time respectively. When natural food is abundant, a feeding frequency of 2 times per day given at 9 to 10 a.m. with one-third of the daily ration and at 3 to 4 p.m. with two-thirds of the daily ration is well and good. In tilapia culture, it is recommended by certain authors that a feeding frequency of 3 times a day is ideal for the 25 to 100 grammers tilapia and 2 times a day for those that is greater than 100 grammars. As we can remember earlier, we have complete feeding and ad libitum feeding in one of the strategies of feeding. So by definition, complete feeding is feeding with food that contains all the necessary nutrients such as protein, fat, carbohydrate, vitamin and mineral, and with sufficient amount of foods required by fish for maintenance and growth. It is also a feeding strategy that is employed when natural food is absent or negligible and where formulated feed is given a day or two after fish stocking until 1 day before harvest. For the ad libitum feeding, by definition, it is giving unrestricted amount of feed until satiation or until the fish are so fully satisfied that they do not consume all the given food within the usual feeding time. So, in complete feeding, it is better to employ the sliding feeding scheme that is from high to low feeding rate. For example, 12 to 10 to 8 to 6 to 4 and to 3% of fish biomass rather than the flat rate like the 4% or 5% or whatever single percentage rate which will be used all throughout the duration of the culture period. The 12 to 10 to 8 to 6 to 4 and to 3% feeding rate scheme had been established to be the ideal for 4-month shrimp culture.
Feeding plays a vital role in ensuring the healthy growth and well-being of aquatic animals. A proper feeding procedure helps maximize feed efficiency, reduce waste, and improve overall productivity of the pond. By following these methods, we can ensure that all fish receive adequate nutrition while maintaining water quality and optimizing feed management. First, broadcast the feeds evenly into the pond bottom. Make sure to spread the feed uniformly for all fish to access it. Feeding them in trays or with feeding area. In shrimp ponds, 6 to 8 is ideal number of feeding trays, 1 meter by 1 meter in size in every hectare. Condition the fish to sounds before feeding. Help fish associate specific sounds with feeding time for better response. Using an automatic feeder. Automatic feeder provides consistent feeding without manual effort. This method ensures uniform feeding and helps fish adjust to feeding sounds, improving their feeding efficiency. Using trays or automatic feeders makes the process more effective. Proper feeding management is crucial for maintaining healthy fish growth, minimizing waste, and ensuring efficient use of resources. The following feeding tips provide practical guidelines on how to optimize feeding frequency, feed size, natural food availability, and environmental factors. By following these best practices, fish farmers can improve productivity while reducing unnecessary feed loss. Feed the fish, not the pond. Only provide feed where fish can eat, avoiding waste. Feeding frequency is the number of times fish are fed in a day. Feeding frequency should be modified according to the size of fish and natural food productivity. Frequent feeding is recommended when the fish are small, when the natural food is scarce, and when the feeds are less stable in water. Increase feeding rate when natural food production is low. When natural food decreases, feed more to meet the fish nutritional needs. Adjust feed size with the size of the fish. So use bigger feeds as fish grow. Broadcast the feed over a wide area. It allows for more fish to avail of given feed, minimizing size variation in fish population and feed wastage. However, there was an observation made that milkfish had better appetite or ate actively when fed in one common but wide feeding tray. This can be explained by the so-called social interactions. Lastly, the ration should be reduced by 25% when the temperature drops to 25 to 27 degrees Celsius and by 50% when either the temperature falls to 21 to 24 degrees Celsius, salinity is over 36 ppt, or dissolved oxygen is less than 3 ppm. Stop feeding when dissolved oxygen drops below 1.5 ppm. Computation involving feeding. The following computations provide a structured approach to calculating feed requirements and efficiency in milkfish farming. Example, you have stocked 50,000 milkfish fingerlings having an average body weight of 3 grams. If these fishes be fed with commercial feeds at the rate of 10% of their biomass daily, how much amount of such feeds would be prepared each day? To find how much amount of such feeds would be prepared each day, we have to follow the formula. Amount of feed per day equals number of fish times average weight of fish times survival rate times feeding rate per day over 100%. Number of fish is the total number of milkfish fingerlings stacked in the pond. Average weight of fish is the weight of an individual fish in grams. Survival rate, the percentage of fish expected to survive during the culture period. In this case, it's assumed to be 100%, meaning no mortality. Feeding rate per day is the percentage of the fish body weight given as feed each day. So given number of fish equals 50,000, average weight of fish equals 3 grams, survival rate equals 100% or 1 since no mortality is considered, and feeding rate per day equals 10% or 0.1. Substituting into the formula, amount of feed per day equals 50,000 times 3 times 1 times 10 divided by 100 equals 150,000 times 0.1, and then the answer will be 15,000 grams or 15 kilograms of feed per day. Next, find for the feeding rate you use with 50,000 milkfish fingerlings having an average body weight of 3 grams and fed 15 kilograms commercial feed each day. For the formula, feeding rate equals weight of feed given in gram divided by total weight of fish being fed times 100%. Weight of feed given per day is the total amount of feed provided to the fish in grams. Total weight of fish being fed, this is the biomass of the fish calculated as number of fish times average weight of fish. Feeding rate is the percentage of the fish total weight that is fed daily. So given weight of feed given per day equals 15 kilograms equals to 15,000 grams. Number of fish equals to 50,000. Average weight of fish equals 3 grams. So step 1, calculate the total weight of fish. So total weight of fish equals 50,000 times 3 equals 150,000 grams. Step 2, apply the formula. So feeding rate equals 15,000 grams divided by 150,000 grams times 100 equals 0.1 times 100 equals 10% or 10% feed. How to estimate the total amount of feed needed for the entire duration of the culture? For the formula, total amount of feed needed in kilogram equals total number of stock times average target weight of fish at harvest in grams times expected overall FCR. Total number of stock is the number of fish that will be harvested. Average target weight of fish at harvest is the expected weight of each fish at the end of the culture period in grams. Expected overall feed conversion ratio, or FCR, is the efficiency of feed usage, representing how much feed is required to produce 1 kilogram of fish weight gain. Step-by-step computation. So given total number of stock equals 9,000 fish, average target weight of fish at harvest equals 250 grams or 0.25 kilograms. FCR equals 1.75, meaning 1.75 kilogram of feed is required to produce 1 kilogram of fish weight gain. Now, apply the formula. Total amount of feed needed in kilogram equals 9,000 fish times 250 grams per fish times 1.75 is equals to 3,937,500 grams, or 3,937.5 kilogram of feed. Since feeds are commonly packed in 25 kilogram bags, we calculate the number of bags needed. 3,937.5 kilograms divided by 25 kilogram per bag is equals to 158 bags. So that's the total amount of feed needed for the entire duration of the culture. Next, how to determine the percentage share of the following types of feed from the total amount needed based on the rate of feed type. So we have starter feed, grower feed, and finisher feed. In starter feed, the percentage rate of feed type is 5%. In grower feed, the percentage rate of feed type is 25%. And finisher feed, the percentage rate of feed type is 70%. For the formula, percentage share of feed type is equals to the total amount of feed needed times percentage rate of feed type. Now, calculate the amount of each feed type. Starter feed equals 3,937 times 0.05 is equals to 196.85 kilograms or 197 kilograms. Grower feed equals 3,937 times 0.25 is equals to 984.25 kilograms or 984 kilograms. Finisher feed equals 3,937 times 0.70 is equals to 2,755.9 kilograms or 2,756 kilograms. So that's how you determine, or that's how you determine the percentage share of the following types of feed from the total amount needed based on the rate of the feed type. How to determine apparent feed conversion ratio, or S value. For the formula, AFCR is equals to total feed given divided by final weight minus initial weight of fish stock. Total feed given is the total amount of feed in kilogram consumed by the fish during the entire culture period. Final weight of fish stock is the total weight of all the fish at harvest in kilogram. Initial weight of fish stock, so the total weight of all the fish at the beginning of the culture period in kilogram. And AFCR, or S value, is a measure of feed efficiency, indicating how much feed is required to produce a given amount of fish biomass. A lower AFCR means better feed efficiency. Example of problem in finding for AFCR or S value. What would be the apparent food conversion ratio if at the end of a 120-day culture period, the fish stocks that had consumed 1,500 kilograms of feed had grown to a total weight of 1,015 kilograms from an initial of 15 kilograms? So, total feed given is equals to 1,500 kilograms. Final weight of fish stock is equals to 1,015 kilograms. Initial weight of fish stock is equals to 15 kilograms. So calculate the total weight gain of fish. So weight gain is equals to final weight minus initial weight. So 1015 kilograms minus 15 kilograms is equals to 1000 kilograms. Step 2. Apply the formula. So AFCR is equals to 1500 kilograms divided by 1000 kilograms is equals to 1.5. So this means that 1.5 kilograms of feed was required to produce 1 kilogram of fish biomass, giving an AFCR of 1.5 is to 1. Stock sampling is a vital practice in aquaculture to monitor fish growth, adjust feed rations, and assess overall health. Proper sampling techniques help ensure accurate data while minimizing stress on the fish, contributing to efficient and sustainable fish farming. So, purposes of sampling. First is to monitor fish growth. So, sampling helps track how the fish are growing over time, allowing you to see if they are developing as expected and adjusting management practices accordingly. Second, to have a basis in adjusting the feed ration. So, regular sampling helps determine the average size and condition of the fish, which allows you to adjust the amount and type of feed, ensuring they get the right nutrition. Third, to check the health condition of the fish. So, sampling provides an opportunity to inspect the fish for signs of illness, injuries, or stress, helping you identify and address health issues early. And lastly, to check the occurrence of disease. So, through sampling, you can detect any outbreaks of disease or abnormal conditions in the fish. So, proper treatments or interventions can be made to prevent spreading. Next, time and frequency of sampling. So, sampling should be done every month and not at shorter periods to avoid stressing or hurting the fish. So, monthly sampling avoids stressing or injuring the fish by not disturbing them too often. Next is minimum ideal sample size is 10% of the stock, but this could be reduced to smaller percentage rate if only to free them from stress. So sample 10% of the fish for accurate results, but reduce this percentage if needed to minimize stress on the fish. And now for methods of sampling. To stupefy or make the sample fish calm for ease of weighing, the following chemical substances may be used. So, 10 to 15 ppm phenoxyethanol or mixture of the following. We have 10 ml, 2 parts of isopropyl alcohol, 10 ml quinaldine sulfate, and 5 ml sulfuric acid concentrated. So use 10 to 15 ppm of the above mixture or 4.5 to 6.5 liters of water per drop of the mixture or 1 drop of the mixture per 4.5 to 6.5 liters of water. So this helps sedate the fish, making them easier to handle without stress or injury. Next, use the mass or book weighing technique and then determine the average weight by dividing the total weight obtained by the number of fish taken as sample. So weigh a group of fish, then calculate the average weight by dividing total weight by the number of fish sampled. For example, 50 fish weigh 330 grams, so average weight equals 330 grams divided by 50 fish is equals to 6.6 grams per fish. So this method provides the average weight of the fish, which can then be used to estimate the total biomass in the pond. Lastly, estimating prawn biomass in the pond. So uh, the formula used to estimate the prawn biomass in pond is the area of cast net is equals to pi r squared. So total weight in grams or in gram or kilogram times total pond area in meter squared divided by area of cast net. So this formula calculates prawn biomass by measuring the area covered by the cast net and multiplying by the pond area. Growth and condition factor of fish. So what is growth? Growth is the increase in size or number of cells or tissues in organisms. In fish, it's measured by body length, depth, or weight. So growth reflects how much the fish has increased in size over time. Growth in fish can be measured using four different formulas. First, absolute growth in grams. So this measures the total weight increase over a specific time period. For the formula, absolute growth is equals to final weight minus initial weight. So, for example, if a fish grows from 10 grams to 120 grams, the absolute growth is 110. So this means that the fish has gained 110 grams over the given period. Second, absolute growth rate gram per day, also known as daily weight gain. This measures how much weight the fish gains per day. For the formula, absolute growth rate is equals to final weight minus initial weight divided by the duration in days. For example, 120 grams minus 10 grams divided by 90 days is equals to 1.222 grams per day. So this means the fish gains 1.222 grams per day. Third, the relative growth rate percent per day. So this measures the weight gain as a percentage of the initial weight per day. For the formula, relative growth rate is equals to final weight minus initial weight times 100 divided by initial weight times the duration of days. For example, um, 120 grams minus 10 grams times 100 divided by 10 grams times 90 days. 110 times 100 divided by 900 is equals to 12.22% per day. This means the fish weight increases by 12.22% of its initial weight per day. Fourth is the specific growth rate, the SGR percent per day, also called instantaneous growth rate. This measures growth based on the natural logarithm of weight. For the formula, SGR is equals to natural logarithm final weight minus natural logarithm initial weight divided by the duration of days times 100. For example, the natural logarithm 120 minus natural logarithm 10 divided by 90 times 100. So the natural logarithm of 120 is equals to 4.78749 and the natural logarithm of 10 is equals to 2.30258. 4.78749 minus 2.30258 times 100 divided by 90 days is equals to 2.4849 divided by 90 days times 100. So the total is 2.76% per day. So this means the fish growth rate is 2.76% per day based on its metabolic rate. Next is the condition factor. So what is condition factor? The condition factor, or the K, is a measure of the relative plumpness or well-being of a fish based on its length and weight relationship. So a fish with a higher K value is considered healthier and plumper, while a fish with a lower K value may be thinner or in poorer condition. So for the formula for condition factor, K is equals to weight in grams times 100 divided by the length in centimeter cube, or length cube in centimeter. So weight, the weight of the fish in the grams, length, the total length of the fish in centimeters, and 100 is a scaling factor to make the value easier to interpret. Example number 1. So a fish weighs 96.92 grams and has a length of 17.75 centimeter. So K is equals to 96.92 grams times 100 divided by 17.75 cube. So 17.75 cube is equals to 5,600.42. So the K is equals to 9,692 divided by 5,600.42. So that equals to 1.73. Next, example number 2. A fish weighs 41.63 grams and has a length of 13.49 centimeter. So K is equals to 41.63 grams times 100 divided by 13.49 cube. So 13.49 cube is equals to 2,452.98. So K is equals to 4,163 and divided by 2,452.98. So the total is 1.70. So for the interpretation, higher K value, example 1, K is equals to 1.73. So fish is slightly plumper. So lower K value, example number 2, which is K is equals to 1.70, fish is slightly thinner. So different species cannot be compared directly because fish with different body shapes have different typical K values. In general, plumper fish, higher K values are in better condition, which can indicate good nutrition, health, and environmental conditions.